What if we could create technology that doesn't just talk to the brain, but becomes the living, learning part of it? What if we could heal neurological damage not by patching it, but by growing new, functional circuits from the inside out? This isn't science fiction. This is the vision behind a groundbreaking project called 𝗕𝗠𝗅. Imagine a world where the line between biology and technology blurs. For decades, we've tried to understand the brain by observing it from the outside, like looking through a window. But the 𝗕𝗠𝗅 initiative, spearheaded at Imperial College London and the University of Oxford, is smashing that window. We're moving from just watching the brain to actively participating with it. So, what is 𝗕𝗠𝗅? It stands for redefining neural interfaces through biohybrid neurotechnology. That's a mouthful, but the core idea is elegantly simple: to build technologies that embody the brain's own incredible properties, its ability to adapt, to change, and to organize itself. Instead of rigid, static implants that the body often rejects, we're creating living, evolving systems that the brain welcomes as one of its own. Think of it like this: traditional neurotechnology is like a guest in your house. It's helpful, but it's still separate. The biohybrid interfaces we're developing are more like a new member of the family. They grow, adapt, and learn alongside the existing neural tissue, becoming structurally integrated and functionally active. How do we achieve this? It comes down to three key scientific and technological aims. First, we're developing programmable biological scaffolds. These aren't just inert structures. They are intelligent, biocompatible frameworks that can be placed in the brain to guide new cells. They act like a smart construction crew, telling cells where to go, what to become, and how to form new, functional circuits right where they're needed. Second, we're engineering addressable cellular systems. This means we're creating cells that we can communicate with and control remotely. Imagine being able to gently guide new neurons to connect with damaged areas of the brain, encouraging them to integrate seamlessly with the host tissue. We're essentially giving these cells a GPS and a mission: find the problem and help fix it. And third, we're designing adaptive control platforms. This is the brain of our operation. These are sophisticated, closed-loop systems that monitor the brain in real time and provide feedback to our biohybrid device. It's a constant conversation. The system can see how the new circuits are maturing and adjust its strategy on the fly, steering the repair process with incredible precision. When you combine these three elements, the smart scaffold, the controllable cells, and the adaptive control system, you get something truly revolutionary: a hybrid device that has the living intelligence of biology and the engineered precision of technology. This creates a stable, adaptive, and truly collaborative relationship with the brain. You might be wondering, why now? What makes this ambitious vision possible today? It's because we're living at an incredible moment of technological convergence. Recent breakthroughs in materials science, synthetic biology, and bioelectronics have all come together. We can now build materials that are conductive, biodegradable, and so biocompatible that they mimic the brain's own dynamic nature. These new technologies don't fight against biology. They grow and adapt with it. This work is fueled by a new, forward-thinking approach to science. Thanks to visionary funding from the Advanced Research and Invention Agency, or ARIA, the 𝗕𝗠𝗅 project operates in an environment that champions high-risk, high-reward innovation. We're encouraged to ask the big questions and pursue bold ideas, free from the constraints of traditional research cycles. This allows us to bring together unconventional teams of experts to tackle science's most fundamental challenges. This project also forces us to completely rethink what invasive means. For years, the goal of neurotechnology has been to be as non-invasive as possible. Implants were often seen as a last resort, but 𝗕𝗠𝗅 flips that script. We're not aiming for avoidance. We're aiming for true integration. If a device can co-develop with your own neural circuits, respond to your body's unique biochemical cues, and actively participate in repair and learning, is it still invasive? Or has it become an embedded, evolving extension of your own nervous system? It becomes something the body collaborates with, rather than rejects. This new way of thinking has huge implications for everything from regulatory science to how we measure safety and success in next-generation medical implants. The potential impact here is enormous. Of course, our immediate goal is to develop new treatments for devastating neurological disorders and injuries like stroke, Parkinson's disease, or spinal cord damage. But the broader significance of rescue is even more profound. We are creating a brand new way to do brain research. By building systems that can integrate directly with neural circuits, we can study the brain from the inside out. We can watch and even influence processes like learning, memory, and regeneration as they happen in real time. This turns our biohybrid platforms into both powerful therapeutic tools and incredible engines for discovery. We're bridging the gap between experimental neuroscience, regenerative medicine, and bioengineering in a way that's never been done before. We are truly standing at the dawn of a new era in neurotechnology, where the boundary between biology and technology becomes a space for mutual learning and co-evolution. The biohybrid systems we're creating at Rescue are not designed to replace biology, but to collaborate with it, to learn from its wisdom, and to mirror its incredible adaptive intelligence. Achieving this dream requires more than just a technological breakthrough. It demands a new kind of scientific collaboration, one that erases the lines between disciplines and embraces a long-term vision. Rescue is the embodiment of that spirit, uniting engineers, biologists, and material scientists to build technologies that are not just precise and functional, but are, in a sense, alive. Ultimately, we are working to redefine how we build, how we heal, and how we understand the brain itself. We're building a future where human and technological intelligence can co-evolve, working together toward a shared goal of understanding and repair. A symbiotic future is not just possible, it's being built right now. Thank you so much for joining me on this journey into the future of neurotechnology. If you found this as fascinating as we do, please hit that like button and subscribe to our channel for more updates on the Rescue project and other scientific breakthroughs. We'd love to have you with us as we explore the frontiers of science. See you next time.